Welcome again in my online session on pharmaceutical analysis. In this session, we'll discuss about how to deal with multiple choice questions. Now, multiple choice questions may be a straightforward question. You can say it is a line pick, or it may be a twisted one, or it may be on the basis of applications of the theoretical things. Let us see one by one how to deal with this. The question is, the region below 200 nanometers is called near UV, far UV, vacuum UV or dense UV. Here this 200 nanometers to 400 nanometers is a UV region, but below 200 nanometers the light absorbs in the air, or air absorbs the light. That's why if you want to work below 200 nanometers, then system needs to be evacuated. So that is called as a system should be under vacuum. So if one wants to work below 200 nanometers, it is a vacuum UV region. This is a kind of a straightforward question. You can see this appears in the literature directly. But even if you don't recall anything, if you don't know anything, then just go by a method of elimination. Here you can say that this near UV is, the term is not there, far UV, this is again term not there, and dense UV, these terms are not there. So the vacuum UV is the answer if you want to go by method of deletion. Okay, let's go for next. A glass cell cannot be used in UV region because it is fragile, costly, transparent to UV or non-transparent to UV. See here, the question is glass cell cannot be used. There is a little twist in this. And why glass cell cannot be used in UV region? Because whatever material of construction used for the sample cell, it should be transparent to radiation which are in use. So the material of construction should be transparent. Here glass cell cannot be used in UV because it is non-transparent to UV. That is the answer. Okay, so fragile or costly or transparent to UV, these are the wrong things. Then if we go for this one, if spin of the electrons in excited state is a parallel, the state is called. This is a typical question of fluorescence and phosphorescence. You can say that when electrons appears in spin paired form, their spins are equal and opposite. Okay, if the electron shifts to the excited state and if its spin changes, if both electron spin is parallel to each other, then this is called as a excited triplet state. This is a metastable state, and followed from excited triplet state to ground state, it emits the radiation, that is a phosphorescence. So this is typically of phosphorescence phenomenon where this situation occurs. Now here the hydrogen bonding shifts UV absorption to longer wavelength, shorter wavelength, better wavelength or half wavelength. Now here if you look at the hydrogen bonding, what happens? The hydrogen bonding stabilizes the molecule. When molecules are stabilized, the electron which are at a ground state, they go still lower energy, and to excite them it needs some higher energy. Okay, so to provide higher energy, the wavelength should be shorter. So here, because of hydrogen bonding, because molecules get stabilized, because electrons at a ground state goes to the still lower energy, to excite it, it needs higher energy, so shorter wavelength. Now next one, which is not suitable solvent in UV region. Now see, what is a requirement of solvent in any kind of absorption spectroscopy? That a solvent should be transparent to that particular radiation. right? So this is a basic requirement of the solvent selection. Now if you look at the list of solvents given in the choices, the water, benzene, methanol and 0.1 normal NaOH in water. Now if you look at this, water is transparent to UV, methanol is transparent to UV, NaOH in water, it is also transparent to UV, but benzene absorbs because it is aromatic system, it is a conjugated system, its lambda max is 255 nanometers, so it absorbs in UV region. So benzene cannot be used as a solvent while working in the UV region, so answer should be benzene. Now move to the next one. What material of construction of sample cell can be used in visible region? Again same thing, whatever material of construction of sample cell, that material should not absorb the radiation. Here when we want to work in the visible region, we have to find out which material is transparent to visible region or which material don't absorb the visible region. So if you look at the glass is transparent in visible region, quartz, it is again transparent to visible region. So glass and quartz both are suitable as a material of construction when we want to work in the visible region. But if you look at the cost, the glass is cheaper than quartz. So glass will work when we want to work in the visible region with the cost effectiveness. But question is, what material of construction of the sample can be used? So quartz can be used, glass can be used, right? So both 1 and 2, because both are transparent in visible region. Moving to the next one. Energy of IR radiation is weaker than UV, weaker than visible, stronger than radio waves, all of the above. Now look, the wavelength range of IR, right? And look, a wavelength range of UV or visible or radio waves. If you look at this, you will find that the wavelength of IR is more than UV, the wavelength of IR is more than visible, and it is lesser than radio waves. And if you look at the relation of energy and wavelength, it is E is equal to hc by lambda. So energy is inversely proportional to the wavelength. So wavelength of IR is longer than UV, longer than visible, so energy is weaker, and it is shorter than radio waves, though energy is stronger. So all three are correct, so all of the above, right? Next one, when two conjugated double bonds present in a ring system, it is called diene, heteroannular diene, acyclic diene, none of the above. Now see, two double bonds in a conjugation system, it is a diene. So it gives the answer. But how others are wrong? If you look at this, why not heteroannular diene? The heteroannular diene means if two double bonds which are in conjugation and if they exist in two different ring systems, then it is heteroannular conjugated diene. But this is not here. The statement says when two conjugated double bonds present in the ring system. Heteroannular diene, the option rules out. Acyclic diene. Acyclic diene means there is no ring system present. These two double bonds are in a straight line, no cyclic structure. But here the question is when two conjugated double bonds present in the ring system. So there is existence of ring system. So no question of acyclic diene. And a diene is a correct one. So none of the above is also wrong. So correct answer is here diene. Moving to the next, absorption maxima of acetophenone by calculation. Now when this question, this is the application based question, you can say that the application of Woodward-Fisher rule. If we know a structure of a compound, then from that structure we can theoretically calculate lambda max of a compound. Now if you look at this, then when it is asked that acetophenone, let's draw a structure of acetophenone here. See, this is aromatic compound where here you can see C double bond and here CH3. Let's bear with this drawing. Okay, so this is a benzene, you can say. Here it is a benzene, carbonyl group and CH3. Now if you look at this, this is a typical aromatic system. So one should recall how to calculate lambda max for aromatic system. Now aromatic system, in that if it is a system like this, aromatic C double bond OX, and if X is alkyl group or ring residue, it becomes aromatic ketone, right? It is aromatic ketone. So the parent value for aromatic ketone is 246 nanometers, right? This is a parent value for aromatic ketones. Now in aromatic system, what we have to see is the substitution on ortho, meta and para positions. So here in this particular structure, there is no any substitution at ortho, meta and para. So there is only basic value, that is 246. So here answer is 246. So this is a typical application based question. If you know Woodward-Fisher rule for calculation of lambda max theoretically, you can attempt this one. Okay. Now moving to the next. See, UV visible spectroscopy is also known as translational spectroscopy, rotational spectroscopy, electronic spectroscopy and vibrational spectroscopy. Now see, when we discussed about spectroscopy, we talked that the spectroscopy technique is based upon what changes takes place with respect to electromagnetic radiation as well as a matter. Here this translational, rotational, electronic or vibrational, these spectroscopies are reflecting changes taking place with respect to a matter when light interact. Now when here in question it is UV visible spectroscopy, it means interaction of UV visible light with the matter. After interaction of UV visible light with the matter, what changes takes place in the matter? So energy of UV visible light is sufficient to cause electronic transition. It is sufficiently high to cause electronic transitions in a molecule who absorbs UV or visible light. So this, after absorption of UV or visible light, there is electronic transitions in the molecule, so which is recorded. So here in UV visible spectroscopy, it is also known as electronic spectroscopy. All rotational and vibrations, these are associated with absorption of IR radiation. So IR spectroscopy is called as rotational and vibrational spectroscopy, and there is no any spectroscopy based upon a translational motion in the molecule. So here the correct answer is electronic spectroscopy, because UV visible radiation is sufficiently energetic to cause electronic transitions in a particular molecule. Moving to the next one. Which of the following compounds show singlet in proton NMR studies? Now when we think of a proton NMR of any molecule, it is associated with absorption by a proton in the molecule, or this singlet or a signal will be dependent upon the chemical environment of a proton and a number of protons on a neighboring carbon. If you look at all these molecules given, acetone, if you look at the acetone, this is your acetone, which contains CH3, C double bond O, CH3. If you look at dimethyl ether, again CH3, O, CH3. Then tetramethylsilane, it is actually an internal standard used in NMR where it contains 4 methyl groups connected to the Si. And if you look at this, all acetone contains 6 hydrogen atoms, 2 CH3 groups, that is 6 hydrogen atoms. Here dimethyl ether contains again 6 hydrogens and tetramethylsilane contains 12 hydrogens, and these all hydrogens are in same environment. So all protons, it may be of acetone or methanol or tetramethylsilane, they will show a single sharp peak, a singlet. So here all compounds should show a singlet in the proton NMR because number of protons present in each molecule, they are in the same chemical environment and they are attached to a carbon where there is no any proton, so no question of a splitting of the peak. A spin splitting is decided by number of protons on adjacent carbon. Here all protons are attached to the carbon to which there is no any carbon adjacent to it containing or bearing any proton. So all are giving a singlet. So the correct answer is all of the above. Acetone, dimethyl ether or tetramethylsilane, they all gives a singlet because the proton in that particular molecules are in the same chemical environment. Next one, during fluorimetric analysis, there is a slight raise in temperature of the sample solution due to internal conversion, vibrational relaxation, external conversion or intersystem crossing. See here, one should clear about the terms internal conversion, vibrational relaxation, external conversion or intersystem crossing. Internal conversion means a excited molecule gives its energy to its neighboring molecule. So there is a transfer of energy from one molecule to another molecule. That is a light energy transfer. A vibrational relaxation means here is a relaxation of electron from or within a vibrational energy levels. And during vibrational relaxation, they release its excess energy in the form of heat. External conversion, the excited molecule gives its energy to a solvent molecule in the form of light energy. Or intersystem crossing, here the conversion of excited singlet state to excited triplet state because of change in the spin of the electron. So here the release of energy in vibrational relaxation is in the form of heat, and because of that heat energy there is rise in temperature of the medium. So answer is here a vibrational relaxation, not internal conversion, it's a vibrational relaxation. Then let's move on to a next question. Which of the following atom is NMR inactive? It's inactive, not active. Which of the following atom is NMR inactive? So what decides whether your atom is NMR active or not? It will be decided by its spin quantum number. It should have a non-zero spin quantum number. A non-zero spin quantum number. So here, if your number of protons and number of neutrons both are even in that particular molecule, its spin quantum number is 0. Here if you look at the atoms given, a carbon 12, the carbon 12 if you consider, here it has 12 atomic number. It means it has 6 protons and 6 neutrons. So both 6 protons, 6 neutrons, both are even figures. So its spin quantum number is 0. If spin quantum number is 0, it is NMR inactive. Simple. So answer should be C. If you look at C13, here its atomic number is 13. So number of protons are 6 and number of neutrons are 7. One is even, another is odd. So it is NMR active. H1, it contains 1 proton and 0 neutrons. One is even, one is odd. So again it is NMR active because its spin quantum number is half. So here is the not case. In case of C12, both are even. So it is spin quantum number is 0. So NMR inactive. So answer should be C12. Moving to the next one. Which of the following solvent is not suitable for proton NMR studies? Now see here again, when we are going for a proton NMR study, what should be the requirement of solvent? It should not contain a proton, right? So solvent which contains proton, it will involve in the absorption and it will interfere with the spectra. Means ideally requirement of solvent is it should be a transparent to the radiations used. Here in NMR spectroscopy, radio waves are a light source, and if a proton is kept in magnetic field and if it's allowed to pass radio waves, it absorbs. So the solvent should be such that which does not contains proton. Here if you look at this CDCl3, it don't have proton. DMSO, it don't have proton. But methanol, CH3OH, it has a proton. So it will absorb the NMR, and which is not suitable as a solvent for proton NMR studies. So answer is methanol. Moving to next. Which of the following molecules are IR inactive? Now again it's a IR inactive. So what is requirement to absorb the IR radiation? This is the prerequisite, you can say. So prerequisite is that there should be a change in dipole and there should have some unnatural vibrational frequency in a system. If you look at the examples, and again one more thing, that symmetric diatomic molecules are IR inactive. Symmetric diatomic molecules means oxygen, O2. It is a diatomic molecule. O2 is formed by 2 oxygen atoms. So it is a symmetric diatomic. N2, nitrogen, it is again a symmetric diatomic. So these symmetric diatomic molecules are IR inactive. So answer should be oxygen. Here water, H2O, it absorbs. Carbon dioxide, CO2, it again absorbs, but oxygen doesn't because it is a symmetric diatomic molecule. So answer is oxygen. Okay, thank you.